Good afternoon, everyone. Welcome to our Autoimmunity and Immunology webinar. My name is Rebecca, and on behalf of Oxford Global, I'd like to thank everyone for joining us today. Today we have Geert Mudde, Founder, Chief Scientific Officer, and Managing Director from S-TIR Therapeutics, who will be presenting on S-TIR technology platform, vaccines based on evolutionary conserved immunological mechanisms for the treatment of both allergy and cancer. After the presentation, we'll have a 10-minute Q&A session. Please submit your questions through the chat option on the bottom of the menu display. The full recording of the webinar will be made available to download on our secured website. The details will be sent to you after the webinar. Without any further delays, I'd like to get started with Geert's presentation. Geert, over to you.
Good afternoon, everybody. Thank you for joining me today on this special day. It's actually my birthday, so I'm very happy for the present of you listening to my presentation. These are weird times, and it could mean that your head is somewhere completely different, or maybe you're sitting at home and don't know what to do, and you're listening therefore. Anyway, I'm going to present our S-TIR technology platform. And at the top of my screen here, you see 3 logos. And that's because there are 3 companies working with this technology, and they're very much related. As you can see here, this is the background of the S-TIR technology. Basically, it was founded by my colleague Christoph here and myself roughly around 2009 when we started with S-TIR Therapeutics as an allergy company. And then in 2013, we opened the platform for oncology, and we also founded together with 2 British colleagues, TYG Oncology, which is basically a development company. The reason why we started in allergy is actually quite simple, because the next slide shows you that this technology platform is a species-specific platform, meaning that the vaccines only work in one particular species, being, of course, humans and non-human primates, because they're so closely related. And the reason why this is species-specific is because we have 2 modules here, one which is called warhead and generic. Every vaccine that we make has this warhead, and there's an immunogen, which is basically the module to which the immune response is directed, and this one is disease-specific. Now, these 2 interactions here are species-specific. So if you have, so basically the human vaccine would react to human CD32 and human TLR9. And that means that it is very difficult to do in vivo experiments with disease models. As you can see, it is actually quite complicated system in where you have 2 modules where you basically have to mix one module with the other, one-on-one, before you get your final complex vaccine. And for that reason, I was looking at the beginning for an animal model that would be so convincing that people would be willing to look into a technology, because they saw that this really had a benefit. And the only animal model that I could find in non-human primates, because that was the specificity that we needed, was House's mite allergic asthma. And that's why we started with S-TIR Therapeutics. I'm going to show you a few slides on that particular model later on. But before we go there, a little bit more about the platform itself. And as you can see here, okay, the title already said that, allergy, oncology, so depending on what you put here, but also, of course, infectious diseases, and of course, SARS-CoV and coronavirus 2 is an obvious candidate as well. And we're looking into making a vaccine together with probably thousands of companies around the world. But if there's somebody in the audience who is looking for a partner, we're happy to work together with you. Now, back to this module. Why did I actually design a technology where you have all these things in one molecule? And that's some basic immunology, and it goes back to a paper in 1994. I was at that time working in allergy on IgE-mediated antigen presentation, IgE receptors, high affinity, low affinity receptors. Now, while I was doing that, Tony Alansevecchia in Italy and his group published a paper in JXMed showing that if you target tetanus toxoid through IgG antibodies, so make complexes, to dendritic cells, you need 1,000-fold less of your tetanus toxoid to get the same T cell response. This is a T cell proliferation response. We saw similar things in IgE, even more convincing. We have even more difference between with and without complex. But basically, this is the basis for our vaccine, because we target the immunogen through a link to CD32, as if it was a complex of IgG molecules. And because of this, we can say that the CD32 receptor is the natural receptor for IgG antigen complexes on dendritic cells. So every time that the body encounters a recall antigen, complexes with IgG are made, and these are presented to the dendritic cells in a much more efficient way. And that's one of the reasons why recall antigens are so much more efficiently countered by the immune system. Now, everybody knows about memory B cells and memory T cells. They forget about this particular aspect. The response starts 1,000-fold faster than when you do not have IgG. Okay, having said that, it's clear that this is beneficial, and obviously this is a natural pathway, so there are no side effects related to this combination. Now, if you combine the TLR9 agonist, which is a DNA molecule, by the way, to CD32, you would expect something similar. It's not quite the same, but it is similar. Because the TLR9 agonist, the CpG molecule, is a sticky molecule, the difference between without and with targeting is not 1,000-fold, but a factor 10. But look at this graph here. Here you see human plasmacytoid dendritic cells and activation through CpG TLR9, and readout is interferon alpha production. Now, first the blue line here. This is the normal way that people use CpG as an adjuvant, which means that you have to incubate the cells for a long period of time, overnight in this case, to get a somewhat of a response, which we call 100%, because this is the normal way that people are stimulating the dendritic cells with this adjuvant. And what you see is that you need roughly 3 micromolar to get a maximum response, which actually goes down if you add more. Now, if you add the CpG to this warhead module, so you link it to CD32, all of a sudden you get the same response with a 10-fold lower amount of CpG. But what is more important is that the maximum response is actually 5 times higher. And the reason for this is that when the CpG is internalized through CD32, it goes in as a wave, and TLR9 receptors inside the dendritic cell will become activated simultaneously in sync, meaning that the signal for maturation, because these cells are immature dendritic cells, caused by TLR9 activation is going at a maximum. And that is important because as soon as the cell starts to mature, it stops picking up antigen or protein or anything from the outside world. It stops pinocytosis. Even receptors do no longer go in. And that means that you can't go higher than this because already enough dendritic TLR9 receptors are activated, and then the cell stops. It doesn't take up anymore. So when you use targeting to CD32, you increase this level, and there is a plateau. Because there is only so many CD32 molecules on the surface, you can't overstimulate. Now, what is also important, I already mentioned it, is that when TLR9 gets activated, it stops pinocytosis. And that means that the cell starts to mature, and any antigen that is outside the cell will not be able to come in anymore. So this happens with a classical vaccine where CpG is the adjuvant. The chance that your CpG concentration, when you inject it into the skin or into the muscle, is actually reaching this optimum concentration is practically zero. It's either too much or too little. And at the same time, as soon as the maturation starts, no more immunogen will go in. So you have to make sure that your antigen is going in at the same rate as the CpG. So you need to combine those. And that's basically what's happening in this complex molecule where you have the immunogen and the TLR9 in one molecule. Each time there is a signal for TLR9, immunogen is already in there. Everything is synchronized by CD32, and you get maximum antigen presentation by an always activated dendritic cell. And that leads to a very strong Th1 cell induction, IgG1 antibodies, and CD8 cross-presentation, CD8 activation of killer cells. So let's go and discuss first the oncology field. The oncology field, which is actually represented by OncoQR here, which has the platform, and TYG Oncology, which is our development company, by the way. In this particular case, the antigen that we put into the vaccine is a tumor-associated antigen or protein or peptide. It can be a short peptide, we have an example of that, or a large recombinant protein, and I have an example of that as well. And the issue with these molecules is they're autoantigens, and therefore the immune system refuses to make an immune response. And that's why in the 90s, very, very bad results were obtained with active immunization against tumors. Okay, by combining these antigens with the warhead, tolerance is broken, checkpoints are downregulated or upregulated, depending whether it's positive or negative, and you get a full-blown immune response. And that is actually able to replace any checkpoint inhibitor or a CAR T cell. You don't need them if you use this system. So a little bit about the current treatments of cancer, specific treatments that is. Of course, non-specific like chemotherapy or radiation are not really a comparison, but we have specific treatments. We have the monoclonal antibodies of the first generation, recognizing a tumor antigen on a tumor, and they mimic B cells that make antibodies. The difficulty is that they recognize only one epitope and the tumor may escape. Now, the second generation monoclonal antibodies has come around roughly 10 years ago against checkpoints. Now, we're no longer targeting the tumor itself, but we're targeting specific T cells, which sometimes recognize the tumor, but in all cases recognize their own tissues. So there is, in some cases, a very beneficial effect for a patient where the tumor is really attacked by these T cells, but in all cases there's an autoimmune disease. And there's no biomarker available that tells you which patient is going to have the tumor effect and which patient does not. And there's the bispecific antibodies and there are the cell therapies. I'll skip them for now, but basically in all cases, it is a single arm mimic. The immune system has evolved over millions of years, and it has resulted in polyclonal immune responses of both B cells and T cells at the same time. Not a monoclonal B cell mimicking thing, not a T cell that recognizes a tumor, but everything at the same time. And that is exactly what we can obtain with our vaccine platform and with our vaccines in a totally antigen-specific manner. So far, in over 60 non-human primates with 3 different vaccines, we have not seen a single side effect, despite tremendously strong immune responses that I'm going to show you in the next slides. So this is the mode of action of the S-TIR technology platform in oncology, and later you will see how it works in allergy, and there is some similarity, of course, because we're basically talking about exactly the same activation mechanism of this PDC. The PDC is one of the 2 cell types in humans where there is a constitutive expression, high level, of TLR9. In the mouse, it's completely different. It is in many different cells. These cells express CD32, and that's where the vaccine binds. It's actively internalized, and inside the cell, TLR9 is activated, and the PDC now starts to present the immunogen, but also the warhead, the protein here, on class 2 molecules and on class 1 molecules because of cross-presentation. And the T cells recognizing the immunogen, per definition, are autoreactive and anergic, tolerant. But now for the first time, they see this immunogen by a fully activated dendritic cell. And as a result, they become completely activated. All the negative checkpoints like PD-1 and you name them are downregulated, and the positive ones like B7 are upregulated. As a result, this is a fully operational cell that can help a cytotoxic T cell to kill a tumor. Because also the cytotoxic T cells are anergic in these animals, or in everybody who has tumor-specific cells. They're always shut down. The other cell that I already mentioned that is available in the human immune system is the B cell that also has TLR9 expression, and also the B cell, recognizing the autoantigen that is in the immunogen, is an anergic B cell, and it will not respond. But now, for the first time, it becomes co-activated by the B cell receptor, CD32B, and the TLR9 receptor. And I know that immunologists immediately will say, well, this is a negative feedback loop. True, but because TLR9 is activated, not only the negative checkpoints on the cell surface are down, but also this negative checkpoint is overruled. This B cell is now fully ready to respond to any T cell that recognizes any part of this vaccine on class 2 molecules. And as a result, it will start to make IgG antibodies, and not just a few antibodies, but tons of antibodies. You've never heard more antibodies than from this system. Now, depending on what your immunogen is, these antibodies have a different function. They can neutralize a growth factor. I have an example of that. They can recognize a tumor cell surface antigen, kill by complement. We have an example of that. They can activate NK cells. We have an example of that as well. And of course, the cytotoxic T cells. So basically, one vaccine activates everything nature has given us to combat tumors. And we call that natural antigen-specific checkpoint control, because we downregulate the negative ones and we upregulate the positive ones. And if we stop immunizing, the immunogen that is always in your body will downregulate everything again, and you will not have an autoimmune disease. I have an example of how we can show that. So let's go to the first example, which is called OncoQR 200 or TYG 200 because it's in development by TYG Oncology. The antigen of our first example is HER2/neu, well known by everybody. It is overexpressed on breast cancer, and it is the target of the first generation monoclonal antibody Herceptin and Perjeta, and that's exactly the gold standard that we're going to use to compare the immune response that we induce with the antibodies that have been so beneficial in the clinic. So it is a recombinant protein which we produce, and we add an alpha helix. I didn't mention that before, but there's an alpha helix on this side and an alpha helix on that side, and they two together form a so-called coiled coil, which then gives you this complex. And the affinity is 10 to the minus 10, extremely specific and very stable. So the only thing you need to do is to put this alpha helix on the N or on the C terminal of this protein, it doesn't matter. And then you mix one molecule of immunogen with one molecule of warhead to get one molecule of vaccine. And here's the result. Now, something very important, when we immunize monkeys, we do not use the human autoantigen, we use the monkey autoantigen, because that's exactly the mistake that was made in the 90s. Human proteins were injected into mice. The mouse made a fantastic immune response against the human protein. They went to the clinic and there was no response at all. So we did not make that mistake. We used cynomolgus HER2 in the cynomolgus OncoQR 200 vaccine. So here you see the immune response after 3 immunizations in purple, and in red is the cross-reactivity of these antibodies to the human antigen, which is necessary because if we do an in vitro killing of tumor cells, there are no tumors in these animals, so I have to do it in vitro. I use a human cell line which expresses human HER2. So I can't make use of these huge titers. I have to do it with these lower titers, yet it still works nicely. And how much is this? I mean, this is an artificial arbitrary unit here, but in fact, here after roughly 2 months, we have more than 200 micrograms of IgG with this vaccine at this particular dose. When we go one dose higher, we get antibody levels of roughly 1 milligram, so 5 times higher at the same time rate. Now, you can ask yourself the question, are these antibodies clinically relevant? And here is the answer. The only way to test that is to show that we can compete for binding with Herceptin and Perjeta. This is inverse clinical development. We do not have an animal model with the tumor, so we go to the clinic. We say breast cancer patients can be treated with Herceptin and Perjeta. So if I can show that these monoclonal antibodies are in my induced immune response, then this is more or less a guarantee that at least the antibody side of the vaccine is functional, is working. So here you see the same curve that you saw before, and you would expect that where the titer is highest, that you have maximal inhibition of these antibodies for binding to the natural antigen. But that's not the case. You see when the titer is already going down pretty much to 50%, This is where we have the maximal inhibition. And that means that we have here a lot of antibodies with potentially lower affinity. And here we have less antibodies with higher affinity, better quality. And that is called affinity maturation. And now you wonder, I didn't immediately go to this, why does this titer go down? This is a vaccination. So why does it go down? Well, basically, what is happening is that, first of all, these antibodies are binding to tissue expressing HER2. So that's one way why the antibodies are going down. But in addition, without the pressure of the immunization, the antigen is presented by normal dendritic cells and tolerance is reinduced. That means that we have to, at some point, re-inject the antigen to get the response back up. And I will show you that in a later slide. So it is important that it goes down because we have activated B cells, we have activated T cells, and the antigen is present. That would lead to an autoimmune disease, but it's not the case. So without pressure of the immunization, tolerance is coming back and there is no autoimmune disease, but only an autoimmune reaction. Okay, this is the B cell side of the response, and here is the T cell side of the response. We have generated from each individual monkey autologous HER2 expressing target cells, and you see that 3 days after the immunization, we have a fantastic killing experiment, a killing of 50% of the target cells, and that boils down to that each cytotoxic T cells that is in that well is able to kill 50 to 500 of these target cells. So that's quite a remarkable stimulation. So we have shown that we have antibodies that mimic Herceptin and Perjeta. I'm not going to show you the complement activation and the ADCC. The monoclonals can do ADCC, but they cannot activate complement. The polyclonal serum that we induce is also able to induce complement. So we have all mechanisms of action available with this vaccine to kill tumor cells in vitro. The other example is TYG100. This one is against pancreatic cancer, and the antigen is a short peptide of only 8 kilodaltons, and it is gastrin, and that's a growth factor for pancreatic cancer cells. I will skip the biology of this molecule, but it is an important molecule which has been in the clinic in over 1,000 patients, and patients who actually were able to make an immune response, which were not many. They lived almost twice as long as patients who did not. So it is an important target. And the only point is that you have to induce the immune response. So the previous vaccine was able to induce a minimal response of 20 units, and I'll explain that later, in roughly 40% of the patients. And the next slide shows you that with this vaccine, here you see the 1,000 unit border. So that is 50 times more than what was clinically achieved with the previous vaccine, is already reached after 2 weeks or 3 weeks with a very low dose in every individual animal. But maybe more to your imagination is the following. Here, 14 days after the first immunization, with the high dose, this is 150 micrograms of vaccine, that's what we call a high dose. 14 days later, we have already 40 micrograms of IgG against a single epitope vaccine. And 1 week later here, after the second immunization, we reach 150 micrograms. Now you can go into the literature and look for vaccines like tetanus, diphtheria, toxoid, very immunogenic, very well tested. And you will find that after 3 immunizations, over roughly a year's time, the responses are around 90 to 100 micrograms. And here within 3 weeks, we already have 150 micrograms. That's an amazing amount of antibody. And the reason is that with this particular dose, all those autoantigen reactive cells that are silenced but circulate your body are activated at the same time and start to make antibody. And also here you see that over time, the titer goes down. And that means in this particular case, thinking about that we want to have always roughly 100 units in relation to their previous clinical trial, so 5 times more than what they were able to induce. We need to immunize maybe 6 or 7 months later. meaning we can treat these patients 3 times in a period of 1 year. Okay, now this is a crossover study where we used animals immunized first with the HER2 vaccine and then with the gastritic vaccine and then again with the HER2. And here you see the immune response against the HER2. You see the immune response after 3 immunizations. You saw it until here. Here we gave a 4th immunization. The immune response is immediately back up. We allowed it to go down to completely zero, really zero, 1 shot, and it even goes higher. And at this stage here, we started the other antigen. But what happens is that from that moment on, antibodies are raised against the 2nd immunogen, but not against the 1st one. There is a total antigen-specific interaction for each individual vaccine. Although both vaccines contain the same warhead, there is no crossover when it comes to, or there is no bystander effects. It's all antigen-specific. And in each case, we can turn on the checkpoints or switch them off, on, off, whatever you like. And then the question usually comes before you ask it, what about immunogenicity of the warhead? Are you raising neutralizing antibodies? Now, before I answer that question, the monkey G17 does not have a T-cell epitope. So there will be no response at all if there's not a carrier that can activate the T-cells. And the carrier is the warhead. The warhead is immunogenic. And here is the IgG antibody response against the warhead. And despite high titers of IgG at the moment of activation, you see that the specific response is immediately induced. So yes, there are antigen-specific antibodies against the warhead, but they are not neutralizing. In fact, they can be used as a biomarker for successful immunization. Okay, this is oncology, and I'm now going to switch to allergy, which was actually the first start of our experiments. So here you see the same platform, the same warhead, the same disease-specific immunogen applicable for allergy, and in this case, we're only looking at T-cell epitopes. Now, why am I only interested in putting T-cell epitopes in my immunogen? Why do I not put complete antigens in my vaccine for allergy? And that's very simple. I'm going to take you back to school book immunology, especially the lower part here. You have all learned that IgE antibodies are central for allergy symptoms. They bind to mast cells. High affinity IgE receptors, co-cross-linking of the 2 receptors will lead to histamine release and all kinds of things, and that will give you the antigen. Now, my claim to fame, if you like, is actually in 1990, where I could show with my group, or the group with me, whatever you like, that the IgE is central for allergy induction because also antigen-presenting cells have high affinity IgE receptors. And that leads to a very efficient antigen uptake and presentation. And because these cells are in the skin, for instance, and are immature, they induce Th2 cells, which by then will make IgE switches and they go back. So basically, this is the starting point of my research when I was a young scientist. And then in 1995, I could also show, this is immunology today, but this is also from a textbook by now, that the IgE binds to CD23 on B cells. And that leads to a spreading of allergy from one antigen to the other. I'm not going to explain it, but just Google my name, 1995 Immunology Today, and you will find the paper where this is explained. So IgE is extremely important in allergy, but it is also able to induce anaphylactic reactions because the complete antigen will degranulate mast cells. And that's why you don't want to go with high antigen titers, allergens into allergic patients. There is a direct relationship between the IgE titer and the manifestation of disease. Healthy controls, everybody has IgE a low level. Hay fever patients have a low level as well, roughly maximally 700 units. The allergic asthma patients are in the middle, and atopic dermatitis patients have incredible amounts of IgE. And as a result of that, all treatments which are anything like antigen specific are focusing on hay fever and maybe a few allergic asthma patients, but definitely not the atopic dermatitis patients. Even Xolair, a monoclonal antibody that neutralizes the IgE, is not going over 700 units. And one of the reasons is in order to make these patients respond, you need to produce so much of this monoclonal that every capacity in the world is going to make antibodies for that particular antibody. Because this is so much, and you need to neutralize 95% of the IgE for an effect. Now, the vaccines that we make are totally independent of the IgE level because the IgE does not recognize the vaccine. There's only T cell epitopes in there. So, yes, IgE is important, but more important is the IgE induction by Th2 cells. You could go for Th2 cells, but even these cells are innocent. They just listen to signals from dendritic cells. So what we decided is we go to the plasmacytoid dendritic cell and we tell this cell to make Th1 cells. We don't give the immune system a choice. Every response that we induce is a Th1 response. And if you do that, you will induce IgG antibodies against your allergen, and you will no longer be allergic because there are three necessary things for allergy. In order to get allergy, you need the presence of an allergen. If you go to the South Pole, you will not be allergic. You need the presence of IgE antibodies. Without IgE antibodies, you're never allergic. But you also need the absence of allergen-specific IgG. Because if there's sufficient IgG, the IgE is counteracted. Okay, having said that, this is the aim. And here is our first antigen vaccine that we made. It's called SG100, and it contains house dust mite antigens. Because there was this animal model, the only one that is published, house dust mite induced allergic asthma in non-human primates in the United States. Because that animal model was available, our first vaccine was house dust mite, which is the most difficult allergen because it is the most important one. 50% of all allergics is house dust mite allergic, but it has 25 different proteins. And how do you put 25 proteins into one vaccine? It's not possible, but we did something intermediate. So what I did is I created a recombinant artificial protein containing 29 different T cell epitopes of 10 house dust mite major allergens. And that means that this is a completely new structure that the IgE from the patient does not recognize, but it also means that antibodies raised against this part will have no therapeutic effect. They're only a biomarker for successful immunization. And here is the mode of action. You have recognized here the PDC, the CD32, binding, internalization, activation of TLR9. Now this dendritic cell will produce the Th1 inducing cytokines. T cells recognizing this vaccine will recognize the epitopes from house dust mite, and they are Th2, they're allergic because these are patients with allergy. And due to the fact that these cytokines are produced and this cell is activated at the same time, these cells will switch from Th2 to Th1. And that is the basis for our vaccine. But it also means that the patient needs natural contact with the allergen so that these Th1 T cells can actually switch the B cells from IgE to IgG. Okay, here is the immunogen. You see here the antigens that are included in colors. DERP1 and DERP2, house dust mite, are the most important allergens. Therefore, there are more epitopes from those, and there are a number more. You don't have to look at this, but this is basically the immunogen that we used. And we went into this phase 2-like study at the East Carolina University, animals suffering from more than 10 years of house dust mite-induced allergic asthma, with IgE titers roughly in the level of the atopic dermatitis patients, huge titers of IgE. And we measure early asthmatic responses due to house dust mite, or late asthmatic responses due to house dust mite, or 24 hours later, methacholine-induced, early and late phase. Those are the measures that we take. And we decided that if animals are positively responding, or let me put it this way, significantly have reduced responses in either of these measurements from the house dust mite and or the methacholine in 3 out of 4 parameters, that there is a significant response. And in fact, this is what happened. Professor von Scott, who has been working with this model since 1998 with all big major pharma, big pharmas, says, and he gave us this statement, SG100 is the first and only product that induced significant clinical improvement in both early and late asthmatic responses in 5 out of 8 animals. In addition, this vaccine was tested under the toughest conditions ever. He told me that when I said after the treatment, we have to expose these animals to natural house dust mite, he said, you're crazy, you're killing your experiment. And I said, I need to do that, otherwise my B cells can't switch. We did it and it worked. So there's another very important aspect that I have to show you here, because the warhead and the immunogen of this particular vaccine contain naive B cell antigens. There is no B cell that ever saw a warhead of an immunogen. Yet the immunogen contains only Th2 T cell epitopes. Yet under no condition ever was there IgE found in these animals. So even naive B cells stimulated by potentially Th2 cells did not make IgE, only IgG. Okay, this is what we have done in non-human primates. The next step is that we did something about the species specificity. I've been presenting these data to Big Pharma, and they always wanted to show how can you cure a mouse with a tumor, and I couldn't. And I said, I will not be able to do that. Finally, I decided I can be stubborn, but I don't get anything out of that, so I adapted our technology to work in the murine immune system so that we can have access to mouse models, but at the same time to the canine dog immune system for pet animal health, and sheep so that we can use big sheep for difficult peptides. That's basically the idea. But first, let's talk about the marine immune system. When I started with this, the people at the grant giving offices in Austria told me, why don't you make a mouse vaccine? And I said, I don't, because there is so much difference between mice and humans. There's only 70% homology between the two immune systems. And importantly, TLR9 is expressed in the mouse in practically any tissue, whereas in humans, it's only in dendritic cells and B cells. So the chance is that the mode of action is okay, but I might get all kinds of side effects, and I don't want that. Now, after having treated more than 60 non-human primates, I know there are no side effects, and I felt it safe to go to the murine immune system. So we changed this part so that it recognizes murine CD32 and murine TLR9. And whenever we did a autoantigen, so basically an oncology experiment, we used the murine version of the antigen. And here is the first example. So OncoQR 200, that is the HER2 specific vaccine, was created and immunized into BALB/c mice with a standard concentration of vaccine. And you immediately see that there is a very nice IgG1 immune response against the autoantigen murine HER2. Here we also used a carrier, so basically the same vaccine as the blue curve, but without the CpG. And you would expect that there is no response at all against the autoantigen, but there is some response in some of the animals. It's not in all animals. This is 6 out of 6, and this is maybe 2 out of 6 or 3 out of 6. It means that BALB/c mice are prone for autoimmunity, because this means that you can break tolerance just by injecting it with a carrier. But what is more important from this one is actually the IgG2A class switch, because BALB/c mice are Th2 mice, which always make IgE and never IgG2A. And here is the result. The carrier is not able to induce any IgG2A, which is the Th1 version in the mouse, against the autoantigen, but the classic vaccine with the CpG is very well able to do that. So we have been able to show that we can get very fast and high antibody titers with a murine vaccine against the autoantigen ERBB2 or HER2. Antibodies the same as in the monkey. And here is the T cell immune response. So we immunized animals, and you see immediately that when we test T cells, CD8 cells, against autologous targets expressing cytoplasmic HER2 or ERBB2, there is no killing in the CD8 positive nor in the CD8 negative fraction. But 14 days after the third immunization, when we sacrificed the animals and we took the spleen cells, we have a very significant killing in the CD8 cells, but not in the CD8 negative cells. And this basically confirms what we have demonstrated in the monkey. These levels are very much comparable when you calculate the target to effector cell ratio. Now, having said that, there is of course also the immune response against the alloantigen, which is maybe also important for when we go to allergy or when we go to infectious diseases. And here you see the immune response from a number of vaccines that we tried against the carrier. This is the carrier, which is part of the warhead, or was also the carrier, which was a part of that control vaccine. Here you see vaccines at different concentrations, either used as TYG100 or OncoQR200, or this control carrier. So the green and the purple curve here represent the same as what you saw here, the blue and the red one. Okay, having said that, so we immunize with these vaccines, but their warhead is always the same, so we can look at the immune response against the warhead part. And what you immediately see that despite a dose difference, the IgG1 response is actually quite the same. And in all cases, much stronger than the carrier. But if you look at the IgG2A response, there is a dose-dependent induction. The carrier, like it did not induce IgG2A for the autoantigen, also does not induce IgG2A against the alloantigen. But now the low dose, the middle dose, and the high dose give increasing levels of IgG2A antibodies against this alloantigen, this foreign protein. So where the total IgG is not really influenced, the IgG2A is. So class switch is directly dependent on the amount of CpG that is actually in the vaccine. And that could be very important when we switch again to allergy models with mouse, where if we give a higher dose of vaccine, we have more CpG and a better chance of inducing IgG molecules and getting rid of the IgE there. So I'm getting to the end of my presentation. I'm in conclusion now. I can say that TYG100 induces very clinically relevant titers against a very small molecule called gastrin within 10 days of immunization. Clinical trials with the predecessor showed that you needed 16 units. That's always generated within 10 days after the first immunization. You can combine this vaccine with gemcitabine. I did not show you the data. It delays the response by 1 week, so you're still at that clinically relevant titer within 14 days. Also, TYG200 or OncoQR200, whatever is the name, induces polyclonal B and T cell responses. We have cytotoxic T cells there in both mice and in non-human primates. And we have complement activation, ADCC, and growth factor starvation. I didn't show you the details. Again, contact me after this presentation, and I'm happy to show you all the individual data behind this. This is just an introduction into the technology. So we can say that the species-specific human or mouse S-TIR technology is now confirmed in 2 non-related species, which are actually evolutionary quite distant from each other. And that means that this mode of action is very well conserved in evolution, meaning that the step from non-human primates to humans is a minimal one. There's only 3% genetical difference between the 2 species. So, S-TIR enhanced vaccines induce unprecedented Th1 immune responses against low and non-immunogenic targets, induce polyclonal B and T cell responses, and the mode of action is evolutionary conserved. And there are no side effects either in non-human primates or in mice, despite TL9 expression in multiple tissues. And this is the patent portfolio. And now I'm ready for questions. Thank you very much.
Thanks, Geert, for this presentation. Now I'd like to open the floor for discussion. Remember to send your questions through the chat feature of the platform. So for our first question, what is the advantage of using a 2-module system? Why not produce a single recombinant vaccine comprised of WH and immunogen?
Okay, yeah, that's a question that, of course, I've been asked several times, especially at the beginning, and there are several reasons. If I go back to my picture here, You see here, this is the protein of the warhead, and here is the CpG. This is conjugated by a chemical linkage, and it goes to lysines in the molecule. And in fact, lysines are, maybe I should go to the first presentation. If I go here, you see here this alpha helix. This is actually where the CpG molecules go. So if I would produce this molecule as a single protein and then I would conjugate the CpG molecule, it would go all over the place, including to the immunogen. And that's something you don't want because you need your three-dimensional structure to be intact, especially in oncology. In addition, every vaccine that you make will have different difficulties in production levels. So the fact that you put an immunogen to this single chain, if it would be a recombinant protein, would mean that expression levels would be very, very much different. And by having a single cell line available for this protein and a single method for coupling this CpG molecule, everything can be produced under GMP, stored, and this is a very stable molecule. It's stable for more than multiple years. You can produce that, and every time you have a new immunogen, you just add them to each other. You can optimize production of this part independently of the warhead. And if it's a peptide, you just order it. You don't produce anything, you just order it. And you can start immediately to make your vaccine. And if this is a recombinant protein, of course, then you have to make a cell line or you do it in bacteria. Everything is possible. But you have to only concentrate on this part of your new vaccine. Whereas if it is one molecule, you have to reinvent the wheel of producing it and coupling the CpG to it. So it looks complicated, but it is the better way. Thank you.
Okay, for next question, your technology seems to be very powerful, but is it not complicated to produce a vaccine?
Yeah, that fits nicely to my previous answer. So yes, it is. There are multiple steps. As I already indicated, this is a recombinant protein. It's a single-chain molecule antibody, a single-chain antibody, and we have a cell line that produces this. So you purify this single chain, and then you have to couple the CpG that is synthesized chemically. That's a two-step situation. You have your module. You can store this. You produce your second module, the immunogen, and you combine the two. So there are multiple steps, and that looks complicated, but it is easy and very simple to make. Every student that came to our labs was basically able to reproduce the whole procedure in his first experiment. So once you have your molecule under GMP, you don't have to bother about that anymore. But process development takes a little bit longer than what you're used to. But again, as I said, it's not a difficult thing to do, and we have very high yields. So with one single batch of 20 liters of this single chain, we can produce so much warhead that we can store that for 3 years to serve the world.
How long would it take to prepare a new vaccine on your platform from design to in vivo POC?
Yeah, POC is proof of concept, in vivo proof of concept, I guess. So yeah, maybe the best example is the OncoQR 200 vaccine, where this particular immunogen here is a recombinant protein, meaning you have to design a plasmid with the gene for that particular protein and the alpha helix either on the N or the C terminal side. We did that in August, and we produced the molecule, we purified the molecule. In the meantime, of course, we prepared warhead, we mixed the two, and we made our vaccine and injected non-human primates with that. And in April, we had in vivo proof of concept. That means from design on the blackboard or the whiteboard to in vivo proof of concept for this rather complicated molecule because of recombinant production, 6 months. If you would take this as a peptide, so let's say coronavirus, we use a peptide that is able to induce neutralizing antibodies. We order that peptide, we will have it in 3 weeks, and your vaccine is done in 4 weeks because it doesn't take much time. And we will have in vivo proof of concept if we go to mice in maybe 2 or 3 weeks. So that could be 2 months, depending on your antigen.
Okay, next question. Could you please explain why It is PDC and not the other DC or macrophage to convert Th2 cell to Th1 cell.
Okay, yeah, so the natural cell expressing TLR9 in humans is the PDC. Monocytes, macrophages, well, you can find RNA there, and under certain stimulation conditions, you upregulate the TLR9, but under normal non-disease conditions, the TLR9 is only constitutively at high levels expressed in PDC. So that is the target cell that we're aiming for. And it turns out that if you activate a PDC by TLR9, it produces Th1 inducing cytokines. And these Th1 inducing cytokines will switch the Th2 cell to Th1. So it is the target cell of choice. Now, normal dendritic cells and monocytes also express CD32. So the vaccine will also go in there and it will stimulate under normal conditions, as it always does. But very likely the PDCs, the monocyte dendritic cells or whatever, will be in the same germinal center, and the interleukins produced by the PDC in that germinal center will have a major impact on the other cells. They will work together, and both the normal dendritic cell, the Mo dendritic cell, or the PDC will stimulate T cells, but always in the environment that will induce a Th1 response. I think this was nicely demonstrated even in the mouse, where TLR9 is expressed in many more cells.
Thank you very much for your questions, and once again, Geert, for this excellent presentation. On behalf of Oxford Global, I'd like to thank everyone for joining us today. I'd like to say a big thank you once again to Geert, who has had a lot of time making this webinar as interesting and as valuable as possible. If anyone has any further questions, please do email them across to my colleague using the details on the screen. All that remains to be said is have a great day, and we look forward to welcoming you to one of our future events.